Hello, my name is Alec Phelps. I'm a junior biology major, and I've been participating in neuroscience research with Dr. Kevin Daly's lab at the WVU Department of Biology. A corollary discharge circuit, or CDC, is a neural pathway from motor control centers to sensory and motor neuropil. CDCs have been observed in nearly every organism and sensory modality studied to date. They typically function to disambiguate internally from externally derived stimulus sources in order to elicit an appropriate behavioral response to the environment. The best standard for understanding CDCs is in the cricket's auditory system. A corollary discharge interneuron sends a copy of a motor signal to the auditory areas, which deafens the cricket during their stridulation song. This is represented in figure 1a. This CDI allows the cricket to remain attuned to the auditory environment during its song. However, the CDI projects to many more areas than what is discussed as a part of the cricket CDC, as seen in figure 1b, which other functions are mostly unknown. Drosophila melanogaster has a CDC called ascending histaminergic neurons, or AHNs, located in the ventral nerve cord, or VNC. The AHNs and other types of neurons can be reconstructed with connectomic software project known as CatMade. Morphological identification of AHNs and their synaptic partners will provide a much better understanding of the CDC mechanism. The goal of this project is to morphologically identify AHNs in Drosophila melanogaster and exclude all other potential neurons that do not correspond to AHN morphology, and finally identify pre- and postsynaptic partners. This morphological identification is achieved through serial section transmission electron microscopy data combined with female adult nerve cord connectomics tools in CatMade. These connectomics tools help recreate 3D rendering of neuron morphology by tracing cells deeper into the VNC microscopy data. This reconstruction of Drosophila neurons in three-dimensional volumes help classify the neurons by structure, as well as labeling synaptic input and output of different partners. Potential AHN candidates that are found are then traced. Candidates that do not exhibit all morphological characteristics, such as a corkscrew dorsal projection followed by lateral projection to the brain, are identified and annotated as not AHNs. Synapses are identified by locating electron-dense vesicles along the cell borders, which is shown in figure 2b by the red square. Synapses are either labeled as presynaptic or postsynaptic. Presynaptic partners synapse on to the AHNs receiving input. In figure 2a is how CatMade denotes this with blue arrows. While postsynaptic partners are synapsed onto by the AHNs, providing the output. Synaptic partners are then classified as ascending, meaning the cell body is located in the VNC, descending, the cell body is located in the brain, or local, which means they stay in the specific neuromere. Figure 2c shows what a synapse looks like in the 3D reconstruction. Resulting from this neuron tracing and reconstruction, we have identified 4 AHNs that can be seen in figure 3 and figure 4a. The yellow pair is the mesothoracic AHNs, which project to the front 2-leg neuromeres in the brain. The blue pair is the metathoracic AHNs, which project to all 3 neuromeres in the brain. 4a shows a sagittal view of the VNC and AHNs inside Drosophila. To confirm that these were in fact AHNs, we traced all 165 potential AHN candidates that did not exhibit morphological characteristics, which can be seen in figure 4c, as well as their neuron tract in figure 4b. We also identified several synaptic partners to these AHNs, which can be seen in figure 4d. Synaptic partners that were traced over 1,000 nodes in length from the synapse back are then characterized as ascending, descending, or local. Many synaptic partners are both upstream or presynaptic and downstream or postsynaptic. of the AHNs. Upstream partners characterization can be seen in figure 4E. Thicker lines in this figure correspond to a higher amount of presynapses, the highest being from possible descending neurons, with 69 synapses to date. Downstream partners characterization can be seen in figure 4F. Thicker lines also correspond to a higher amount of postsynapses in this case, the highest being from descending neurons at 49 synapses to date. This provides a stronger foundation for future experiments by identifying upstream and downstream partners to the AHNs. We can then search GAL4 driver line databases for other studies of relationships between AHNs and their specific synaptic partners. All in all, progressing the mechanistic understanding of CDCs in Drosophila melanogaster. Thank you all for your time.